So I'm just going to summarize some of the other sessions at the AACR Advances in Malignant Lymphoma. So there was a really interesting session on looking at sort of the changing paradigms in lymphoma classification. And I think a lot of that has come about with the advent of higher resolution technology. So, if we use diffuse large B-cell lymphoma as a kind of the model lymphoma, it's obviously the most common high-grade lymphoma. And we know that outcomes in diffuse large B-cell lymphoma, although is good for probably about 50 to 60% of patients, there are probably about 40 to 50% of them that will have poor outcomes or may relapse after induction therapy. Now, a lot of these high-resolution technologies, either through either exome sequencing or whole genome sequencing or RNA sequencing, what they're telling us is that actually diffuse large B-cell is a very heterogeneous lymphoma type. I don't think we historically have always thought of the diffuse large B-cell as, very binary in its classification, either as either ABC, activated B-cell type, or GCB, germinal center B-cell type. But we're beginning to see from these studies that have emerged in the last few years that actually it's much more complex than that. We're beginning to see that it's not just a binary classification, but there are 4 or 5, even more different types of classifications within diffuse large B-cell lymphoma, each with its own unique biology, and potentially also meaning that we may need different targeted approaches for each of these different subtypes. So that obviously muddies the water somewhat to the more complex, the more heterogeneous these lymphomas are. How can we potentially target these different molecular subtypes? And I guess that leads on very nicely to the other session that was a very there was very interesting debate in this session looking at sort of how we can, looking at biology-based clinical trials. How can we bring in a lot of the, translate a lot of the molecular information that we now know about most of these lymphomas into clinical practice? And, the title of this session was, biology-based clinical trials, a myth or hope for reality. And I think it's a great title in the sense that there are a number of challenges in bringing molecular information into clinical trials. And it has been exemplified by a number of existing clinical trials. We know, for example, that there have been so many trials trying to improve or augment R-CHOP, which is a standard induction therapy for diffuse large B-cell lymphoma, in the hope by adding in an additional agent, it would improve the outcomes for patients. This is not materialized at present. And it may mean that we need to, rethink the way we design clinical trials in diffuse large B-cell lymphoma, taking into account these different molecular subtypes. I don't think we are there yet, and this was certainly highlighted by a number of studies. But I think that the more we understand about the biology, the more that we interface the biologists with the clinical trialists in the way that we develop new clinical trial designs. And I think that I suspect that we will develop new ways of developing clinical trials for specific molecular subtypes of diffuse large B-cell lymphoma. And that finally led on to, I suppose, a more optimistic session of using liquid biopsies for sort of real-time monitoring of lymphomas. We know that one of the challenges in sort of lymphoma research or translational lymphoma research is that unlike other cancer types like leukemia, where you could basically collect tissue from the peripheral blood or from the bone marrow, in lymphomas you obviously need lymph node biopsies, and the challenge is that to be able to undertake a lot of the molecular studies or molecular analysis that we require would require you to have a biopsy, and we require you to have an excision biopsy. And we know that although that's the gold standard, a lot of that is changing over time. We obviously need biopsies at a faster rate, and so our biopsies are getting smaller. So instead of excision biopsies, a lot of clinical practice now is moving toward core biopsies. And so the challenge with that is that we are getting smaller and smaller biopsies to undertake this molecular analysis. And So, and in comes sort of liquid biopsy, thought to be, a potential surrogate for tissue biopsies. And this was quite an exciting session looking at the potential clinical utility of liquid biopsies, using ctDNA, molecular profiling, circulating tumor DNA that can be detected in the peripheral blood of patients. It's obviously minimally invasive, and it can be serially collected over time as patients are receiving treatment. So what was exciting in this session was that there were a number of speakers that spoke to the potential in this area, either using liquid biopsy as a dynamic monitoring tool in terms of using it to assess response to specific targeted therapy, also potentially using it to as a prognostic tool. So, looking at the levels and quantity of ctDNA levels after patients had received induction therapy, looking at its role, as an interim analysis and end of induction analysis. So I think this is an area where there is a lot of potential of bringing in the biology, bringing in, molecular analysis into lymphoma treatment, and also lymphoma sort of risk stratification, risk assessment, and also for monitoring minimal residual disease over time.